Hello, my name is Ensieh Hosseini. I'm a researcher in University of Glasgow in Bendable Electronics and Sensing Technologies group. And here I present our research about biodegradable amino acid-based pressure sensor. In this presentation, first I talk about our motivation, why developing biodegradable sensors are important and their advantages. Then I will talk about synthesis of amino acid-based biodegradable composite. And later I'll move on fabrication and performance of a biodegradable piezoelectric pressure sensor, and at the end, conclusions and future work. Over the last decade, there have been an increasing demand for development of biodegradable sensors because they offer unique properties. For example, they are biocompatible, they are environmentally friendly and reduce electronic waste. And in addition, they are ideal as temporary medical implant for continuous in vivo body monitoring, and no need for extraction surgery. And usually they have low cost. However, most of developed biodegradable sensors often have weak and unstable electrical performance, and they usually have short lifetime for clinical application. Therefore, applying proper packaging is an important step to achieve a controllable degradation rate. Few examples of biodegradable pressure sensors reported in the literature and are mainly based on biodegradable synthetic polymers such as PLA and PGS, but fabrication of biodegradable sensors from natural and bio-based materials such as amino acids, proteins, and polysaccharide-based polymers have huge advantages due to their natural biodegradability, renewable nature, and low cost. And here we used amino acid glycine as an active layer to fabricate a biodegradable sensor, because amino acids can be self-assembled in organized polar crystalline structure at large scale. Recently, a high piezoelectric coefficient have been found in glycine. Biodegradability of glycine has been well established because it's been used for drug administration, and glycine molecules are able to organize in self-polar crystalline structure with room temperature solution-based processing, and also doesn't require high electric field to induce piezoelectricity in them. And it has high piezoelectric coefficient in beta phase and in some specific crystallography orientation. However, beta-glycine crystals are brittle, and the beta phase, which has the higher piezoelectricity, is thermodynamically unstable and usually co-crystal with other phase of glycine like alpha and gamma phase. But and that's the reason they have not been much explored for sensing device application. But here we could fabricate a composite of glycine with natural polymer of chitosan, and these composites have enough flexibility and is good stable, and that's why we were able to fabricate a sensor from that. For this, first we prepared 1.5 percentage chitosan solution in 1% acetic acid solution, and then a series of glycine to chitosan composite solution were prepared by dissolving different amount of glycine powder in the prepared chitosan solution by stirring, and then the solution casted in a petri dish, and through evaporation technique we could have a flexible film. Here shows the optical image of just plain chitosan, and figure B and C are glycine chitosan composite, and figure D is just pure glycine. I did it just to show as a control experiment that how glycine can have effect on the polymer selectivity of glycine, because here that we didn't have any chitosan, the crystal were mainly alpha phase, and these two we found that 0.8 to 1 is the optimum ratio of glycine to chitosan, because in this ratio we had a uniform film and we had a uniform and enough flexible film. And here is the SCM image of the film with the 0.8 to 1 ratio of glycine to chitosan. It shows that how well the glycine crystal are embedded in the amorphous chitosan matrix, and each of these crystals are like a spherulate with the nucleation point in the center, and then the fibril of a spherulate radiated out from this center. And you can see they are embedded in the amorphous chitosan matrix. And the XRD pattern of the composite confirmed the formation of beta phase of glycine crystals. And we noticed that crystals were very stable inside the chitosan matrix. We made the XRD after a few months. We haven't noticed any phase transformation in that. And we think this stability comes from embedding crystal inside the chitosan matrix. And also there is a possible electrostatic interaction between the polar group of glycine and functional group of chitosan. After preparing film, a pair of parallel electrode of magnesium were deposited on the both sides of the film by thermal evaporation of magnesium and using a hard mask. As you can see in this figure, the magnesium electrode were deposited on both sides of the film, and then it's connected to wire to able to measure the signal. And this video shows the output voltage from the device when it was touched just gently by hand. A repeat compression and release process show the sensor responding uniformly with periodic positive and negative alternation. And then we used a mechanical shaker that generate compressive load at a specific pressure, and the output voltage generate from the device under each applied pressure were measured using an oscilloscope. And this figure shows the output voltage from the device under the dynamic pressure of 5 kilopascal, and the voltage generation is, as you can see, is quite stable and repeatable with fast response time. And this figure shows that voltage linearly increased with applying compressive pressure, and the sensitivity was about 1.42 millivolt per kilopascal in the pressure range of 5 to 25 kilopascal. And this table shows a comparison of piezoelectric sensitivity of the device comparing with other reported non-degradable piezoelectric polymers such as PVDF and PVDF TRFE. Although the sensitivity is lower, but still is in quite good range, and we think the fabrication process can be studied further to control and optimize the crystal structure and enhance the piezoelectric properties. And here we check the in vivo degradation of the sensor by immersing it in a cell culture medium. As you can see in this figure, the magnesium electrode degrades very fast in the PBS solution in just few seconds. And after dissolving electrode, although the sensor needs some time to fully degrade, might be few days, but is not functional. So due to fast solubility of magnesium electrode, we think it's good to use some polymer with tunable degradation rate to package the device, especially for in vivo application. And in this case, biodegradable polymers such as PGS are ideal for packaging as they have a tunable degradation rate. And then the sensor functional lifetime would depend on the degradation rate of encapsulation layer. In addition, using a thicker layer of magnesium or magnesium electrode or magnesium alloys is other way to control dissolution properties of electrodes. And in conclusion, we develop a fully biodegradable and flexible pressure sensor with solution-based process and magnesium electrode deposition. The room temperature simple solution-based fabrication process and biodegradability make the presented sensor an attractive alternative for eco-friendly sensor applications. And the fabrication process has the fabricated sensor has a potential to be used for single-use healthcare applications or implanted in the body after appropriate packaging as a pressure sensor or for electrical stimulation of tissue. And as I mentioned before, we think the fabrication process can be studied further to control and optimize the crystal structure and enhance the sensor performance. And at the end, I would like to acknowledge European Commission and EPSRC for funding this project. And thank you for listening. These are the details of the webpage of the BEST group. If you have, if you can see more about our research activities here, and if you have any question, please contact us through emails. Thank you.